We're in the tissue level of organization today, and I want to go to the book for a second just to get us started on what's going on here. I'm not going to go through the book material exhaustively. I just want to point out some things and highlight what's important so you understand what you're looking at on the microscope today. So in this chapter, it starts off with talking about the types of tissues. There are 4 types of tissues, and actually, come to think of it. Let me just open up my notepad page so I can write this stuff down. There's epithelial tissue, connective tissue, muscle tissue, and nervous tissue. Epithelial tissue is primarily concerned with covering and lining things. So the most obvious epithelium is the outermost layer of your skin, your epidermis. So that's obviously covering your entire body. But individual organs are usually covered by some sort of epithelium. Not all of them, but most of them are. And then hollow spaces within organs are usually lined with some sort of epithelial tissue. There are a lot of other functions for the epithelial tissues, but those are kind of the big picture part of it. Just copy my arrow here. Connective tissue is not really well described as connective tissue. There's a bunch of different tissues that belong to this group, and there's not really one word that sums up everything they do. So to some extent, they are connective. I want to talk about that in a little bit more detail when we get to that particular tissue later on, but that's the best word to put in there. And then muscle tissue is contractile, which means it generates force as it gets shorter, and we use that to move things. And nervous tissue is about communication. It's not the only tissue that communicates, and we'll learn about communication in tissues more later in this course, this semester, and as part of what we do at the beginning of planning E2. But nervous tissue we usually think of as primarily being about communication. Now, I've kind of always presented this material like this. And this is just sort of the broad overview of the types of tissues. It was several years into teaching before I realized that all of the things I wrote up there start with a C. So if that helps you try to remember what each one's about, and just try to think about what the C word was that goes with each one, that's fine. As far as the book is concerned, it just lays out examples of the different types of tissue, points to areas where they're found in the body. But there are the 4 major types of tissues there. Tissues, the types of tissues we're talking about today, or at this point in the course, are really the mature tissues that we see in the adult body. And I mean, really at this level, adult down to child and newborn, the tissues are all kind of established by that point. But the next thing here in the book, which it's getting at, is the embryonic origin of different types of tissues. Early in embryonic development, the embryo is really just differentiated into 3 major regions or types of tissue, which are referred to as the ectoderm. Sorry, I should be pointing at this here. As the ectoderm, the mesoderm, and the endoderm, meaning the outer, middle, and inner layer of tissue. This gives a kind of overview of the different things that develop out of those. When we talk about the nervous system, I'm going to come back to the idea that the ectoderm is the source of most nervous tissue. That's an important part of understanding the nervous system. But also it's the source of the skin, which should be obvious, the outer layer of the body is going to be the skin, obviously. And we'll talk about how the nervous system develops out of that layer of tissue when we get to that part. The mesoderm and the endoderm have various tissues that evolve out of them or develop out of them. Most muscle tissues are mesoderm. A lot of the lining epithelial tissues come from the endoderm. This goes into a little bit more detail. I'm mentioning this partly because it's in the book, and I usually don't actually talk about these layers of tissue until I get to talking about development of the nervous system towards the end of the semester. But it's in the book, so I wanted to point it out. And when we talk about connective tissues, I actually am going to refer to something related to this. So I want to set that up a little bit just to know, so that you know, in the embryo, the tissues are very simplistic, and they're the sources of all these other tissues that'll be fully developed and mature in the adult body. Now, I would normally save this last little thing until the end of talking about the tissues because it's sort of the next step in the progression. We started by talking about atoms and then molecules and then macromolecules and then organelles and cells, and now we're up to tissues. After tissues, we're heading for talking about organs, but between tissues and organs, just like between molecules and cells, there's an intermediate point, which is membranes for what's beyond tissues. Between molecules and cells, we have organelles. Okay? An organelle is not an independent structure like a cell, but it's more than just macromolecules. Membranes are not an individual organ, but they are a little bit more than just tissues. These four boxes highlight the four types of tissue membranes. Really, membranes kind of are where multiple tissues come together. Okay? And part of really understanding that, you need to have gone through the tissues to see what the multiple tissues are about. Three of these tissues, which are three of these membranes, which are mucous membranes, serous membranes, and the cutaneous membranes, those are combinations of epithelial and connective tissues. So at the stage where I've just introduced the four types of tissues, it's not terribly hard to say sort of how those are organized. The fourth type down here, though, is not an epithelial-based membrane. There's no real epithelium involved in it. It's really a combination of different types of connective tissue. So you really need to have heard of the different types of connective tissue to really understand what's going on there. But that's what's called a synovial membrane. It's what lines the joints in your body. And joints are where two bones come together. There are ligaments and cartilage and other things there. So those are all types of connective tissue. And so those are synovial tissues. After the figure here laying out the four major types, it then refers to connective tissue membranes. I should say up here it defines tissue membranes. So it refers to connective tissue membranes first, which is actually the fourth box in the picture above. And then it lays out the three types of epithelial membranes. Again, this is something I would normally save for the end of things, but I want to point it out to you as I'm running through a few things in the chapter because it is at the beginning of the chapter. But it is sort of where we start to think about how different tissues come together to serve as more functional units within the organs we're going to end up studying later on. Then moving on from there, it starts to talk about epithelial tissues. So I want to introduce epithelial tissues and talk about them a little bit. And then I want to talk about connective tissues a little bit. And then we're going to look at these things under the microscope. So let me go back over here to my notes page. Really what we're talking about here in tissues in general is just about how do we classify these tissues. Epithelial tissues are classified based on two major things. First, there are the layers that we might see, and there are two things I need to say about layers, and the other thing is cell shapes. And there are three things to say about cell shapes. As I write these things down, I say two things here, three things there. Just if you're taking notes, I want you to know that you need to leave a little bit of space there. Layers come in two varieties. There are single layers, which are called simple epithelia. A single layer of cells, which is covering or lining something. And then the other type are stratified epithelia. Sorry, I did that wrong. Multiple layers, as opposed to single, which are called stratified epithelia, meaning there are multiple layers of cells. The single ones are kind of obvious. It's one layer of certain cells. Multiple ones, there are multiple layers. There can be different types of cells across those multiple layers, but when we're talking about them in a little bit, I'll explain how we classify them a little bit better. Now, cell shape, there's basically three different shapes we have. There's squamous, which means flat cells. There's cuboidal, which is describing something that's cube-shaped. And if we looked at cube-shaped cell in 2 dimensions, then it's going to be kind of squarish. I say square-ish because it doesn't really look like a perfect square, but that's the best example. And then the third type is columnar, which means column-shaped, or they're tall, thin cells. Okay. So to talk about the layers and cell shapes, I want to move back to the book because that's where we have some pictures to look at. In the book, they start by talking about different types of the functions of epithelial cells. They're usually joined together tightly. So this is talking about the things that attach cells together. But here's where we talk about classification of cells. Now this figure's a little bit odd because it has a bit more information at this point than I really want to get into. But squamous cells are flat. We can see them either in a simple epithelium, a single layer of flat cells, or a stratified epithelium, okay, multiple layers. Cuboidal cells are typically square. We can see them in a single layer or multiple layers. And then columnar cells are tall and thin. Okay. We can see them in a simple or a stratified epithelium. Now the thing that's kind of added in here, and this is not really the place where it's great to put it in because it's going to add a little bit too much information to begin with. There's another type of epithelium that has tall, thin cells. This is called a pseudostratified epithelium. It's called pseudostratified. It's not really stratified. It's actually a single layer of cells, but the nuclei are in all sorts of different positions. The simple columnar epithelia, all of the nuclei are in a row. But here, having them scattered at different heights, under the microscope will make this look like multiple layers of cells. And it's pseudo-stratified. It's not really stratified, but that's what it kind of looks like. Now, the reason why this is in the picture is that these are, in fact, columnar cells. And some sources will refer to this as a pseudo-stratified columnar epithelium. I only ever call it a pseudo-stratified epithelium because, by definition, it's columnar. So that's kind of redundant. There's no such thing as a pseudostratified cuboidal epithelium or a pseudostratified squamous epithelium. They're only ever made up of columnar cells. So that extra word there, columnar, is a little bit superfluous. But really it kind of gets beyond the idea. We're just thinking about how we can put cell shapes and layers together. Now, when we're talking about a simple epithelium, it's always going to be one layer of the same kind of cells. But in stratified epithelia, it's actually possible to have different shaped cells. as these pictures are drawn, they don't really depict that, although the cells at the bottom layer of the stratified squamous epithelium could almost be cuboidal in shape, the way they've drawn them here. And in actuality, stratified squamous epithelia tend to have cuboidal cells down there, or maybe even columnar. When we're talking about stratified epithelia, we name them basically by whatever the top layer of cells is. Okay. The thing is that top and bottom is not a really good way to refer to these cells. Now, I want to show you some actual tissue here. Oops, not from here. I want to go back to Blackboard. At the bottom of the material for this week, there's a link that says University of Michigan Histology website. This takes you to a website with links to all sorts of different examples of tissues. There are two links for each type of tissue. One says WebScope, the other says ImageScope. You want to use the WebScope link. ImageScope is what at the University of Michigan they can use because they have this program on their lab computers, and that won't do anything for us. WebScope, however, is better for what we want to talk about. So I'm going to actually go to a slice of the kidney. The way this works, this is kind of like a virtual microscope. So we have here a digitally captured piece of tissue. Up in the top corner, we can adjust the magnification. So we can zoom in like 4 times, 8 times, 10, 20, 40 times magnification. And we can see the cells up close here. There's always this inset that has the entire tissue on it. So if we want to jump to a different area, we can click here or we can click here and just jump around. Now, this little cluster of cells right here is a good place to kind of start to explain a little bit about looking at epithelia under the microscope. When I turn the lights off here in a second, you'll see a little bit better. Those cells are slightly lighter in appearance than the surrounding cells, so they kind of stick out. That's a layer of cells. They happen to be lining the inside of a tube. It's a very small tube, so they're kind of really clustered together. They are, though it might not seem that way to you, they are in fact cuboidal cells. And I'll explain why that's true in a little bit. They might look, like these might look kind of tall and thin or something like that, but they are in fact cuboidal cells. Now, if we're looking at this cell right here, we can definitely say where I have the pointer is the bottom of that cell, where I have the pointer is the top of that cell. But that works because that cell happens to be oriented to our particular perspective. It's our top and bottom, or how we see top and bottom. But if we go to a cell up here, the top of this cell is where I'm pointing right now, and the bottom of this cell is where I'm pointing right now. Okay. It's sort of upside down and a little bit at an angle. So instead of top and bottom, we use a different couple of words. We use the words apical and basal. Those are just adjectives that refer to the apex or the base of a cell. In this context, it's kind of like talking about a mountain. Okay, the base of a mountain or the apex of a mountain, the bottom of the mountain or the top of the mountain. Except that when we're talking about a mountain, we're obviously talking about the side that's connected to the earth and the side that's pointing up in the air. So up and down kind of makes sense relative to those terms. In a cell, apical is always going to be the side that's next to empty space. Let's go back over here. The empty space in the middle of this tube, where the fluid that is carried through this tube is found, is lined by the top of this entire epithelium. That's the apical surface of that epithelium, apical surface of each of those cells. And then where each cell is adjacent to other tissue, that's the bottom of the cell. Okay. So that's the basal layer of that epithelium, or basal side of that epithelium. Now, I picked this one out because it really stands out as being obviously a single layer of cells wrapped around, lining the inside of this tube. But actually, if we look at these other things that are a little bit fuzzier and harder to make out, we can still say the same thing. So like, there's a little empty space right here in the middle of this tube. It's just off to the side of the one I've been pointing at. And so the edge of the cells lining that empty space, that's the apical surface of all those cells. And then you can kind of see the hint of a line on the other side of each of those cells. That's the basal surface of all of those cells. Okay. So we're looking at a single layer of cells going around that little thing. Now, the one I've been using as an example is probably pretty close to a cross-section of that tube. The second one that I'm showing you, the cells are kind of harder to distinguish, and they don't quite look like cuboidal cells. They are cuboidal cells, but the reason they don't quite look like that is that we probably cut this at a funny angle. Okay, so if you have a straw and you cut straight across the straw so that there's a round edge to either side, that's a cross section. If you cut a straw at an angle so that there's sort of an oval shape to both cut ends, that's more what we're looking at here, kind of an oval shape to this tube cut in section. Now, if we jump around, I just randomly picked another spot here. We can see some more of these more lightly stained epithelial laying around here, but then some of the more darkly stained. And if you start to stare at this stuff long enough, you can start to make out, the empty space in the middle and what looks like a defining line around the outside, even one of these dark ones here. Okay. Doesn't necessarily have to be quite as straightforward as the more lightly stained ones, but we can see that. Okay. And backing off a little bit more, really this tissue is made up all of these type of epithelia. Some of them are lightly stained, some of them are more darkly stained, but they're all just sections of tubes. Some of them are nice round cross sections, some of them are at funny angles. But they're all going to have a little bit of empty space in the middle of them. And the edge of any cell lining an empty space is, by definition, the apical surface of those cells. So here's a little region with a number of pretty good little lightly stained, well-defined epithelia. And so any of these, we can see the clear space in the middle of the tube, and the edge of the epithelia right next to that clear space, that's the apical side. And then just the opposite side of those cells, we can usually see the hint of a line kind of outlining that. That's the basal side. Going back to the picture from the book, okay, we're not seeing any of these wrapped around the space. We're just looking at a few cells that are oriented to our conception of top and bottom. But the top of these cells are the apical surface. On the basal surface of these cells in all these drawings, there's a little thick line that we can see there. That's called the basement membrane, which is not to be confused with the other types of membranes I just pointed out a little bit ago. The basement membrane is really just a very thin layer of mostly collagen fibers that kind of anchor the bottom or basal layer of the epithelia to whatever's beyond it. So as we're looking at these microscope images, when we can see the hint of a little line at the bottom edge, the basal surface of these cells, that's where the basement membrane is. And at this magnification, it's not possible to always see those little lines here, but we can imagine where it would be. Like, okay, here's a really good example. We have two epithelia sort of squished up next to each other, and we can see a pretty good line between them. That's because the basal membrane of each of those is kind of up next to each other, so they stand out pretty well. So imagine if we took two copies of this and put them back to back to each other. That dark line at the basal edge of each of those cells next to the dark line of the basal surface of all of the other cells in the next one, it would really kind of stand out like I just pointed out there. So using the cell shape and the layer classification, we can get 6 types of epithelia. There's actually 9 types of epithelia. And in this picture, they kind of start to introduce us to some of those others. Pseudostratified is another type of tissue. It falls into what I consider a special case for a simple epithelium. So we have these 3 simple epithelia, squamous, cuboidal, and columnar, and then we have this simple epithelium that looks like it's stratified, so it's called pseudostratified. We have 3 stratified epithelia, which are named based on the shape of the apicalmost cells. I didn't say that before. So there's flat cells at the top of a stratified squamous epithelium. There's cuboidal cells or square cells at the top or apical layer of a stratified cuboidal. And then stratified columnar has a layer of columnar cells in its apicalmost layer. In this picture, both layers look columnar. You'll often see that the basal layer is actually cuboidal cells. Now, you won't actually see that because we don't have a good slide of that particular type of tissue, but it actually isn't really just 2 layers of columnar cells. Now, there's a 4th type of stratified epithelia, which is its special case also. It's called a transitional epithelium. And it's called transitional because of what it does. All it does is line the inside of the bladder and then the ureters, which are the tubes that go from the kidney to the bladder. Both of those organs, the ureter and the bladder, have to be able to stretch as they fill with urine. And so a transitional epithelium is an epithelium that's capable of stretching. Now I've switched to a different figure here, which actually lays out more of the types of tissue. So here are our 4 simple epithelia that I just went through, and then here are 4 stratified epithelia. Now we see a picture of transitional. The apicalmost layer of cells in a transitional epithelium are not like any of the 3 shapes that we just saw. They're these big bulbous kinds of things. To me, sometimes they look kind of like mushroom caps or fluffy pillows or whatever you want to call them. But they're big round cells. They don't fit into any of these other classifications. The other cells in the epithelium might look columnar or cuboidal or something like that, but usually they're just kind of weird shapes. Transitional epithelia don't fit into the cell shape aspect, but it definitely is a stratified epithelium. The reason it's called transitional is these cells can be stretched out. So as the bladder fills with urine, part of its ability to hold more urine is that the epithelium actually does stretch out. The cells transition in shape. That's where the name comes from. Now, I said there's actually 9 types of epithelia. And in this figure here, they show us 8. 4 simple, 4 stratified. The 9th type of epithelium doesn't fit into the layers or cell shape classification scheme. And those are what are called glandular epithelia, or just glandular tissue. Glands come in 2 types. There are endocrine glands and exocrine glands. Endocrine glands secrete within the body. Endo means within, crine, secrete. So endocrine means to secrete within. And that's talking about glands that secrete hormones. And then exocrine glands secrete outside the body. So those are going to be the ones that obviously secrete outside the body, like sweat glands or anything in your skin. But then outside your body means something different to your body than it does to you, which might seem kind of strange to say, but it's really an immunological definition. You can put anything in your digestive tract or any other mucus-lined tract. If it stays inside that space, if you swallow something and it never leaves your digestive tract, it might as well be outside your body. Okay? Your immune system doesn't worry about things that are staying within the digestive tract. The immune system starts to worry about things that are trying to get across the lining of the digestive tract or the respiratory tract or the urinary tract or the reproductive tract. So anything inside those mucus-lined tracts are effectively outside the body. So any gland secreting to any of those tracts is considered an exocrine gland. So mucus glands in your respiratory tract, digestive glands in your digestive tract, the glands in your reproductive system, and well, there's not really glands in your urinary system, but anything that secretes across a mucous membrane into one of those tracts is considered an exocrine gland, just like a sweat gland or a mammary gland or a wax gland or something like that. These two types of glands are glandular tissue. The exocrine glands usually have a duct, or not usually, they always have a duct. And so a duct is going to be a layered epithelium. Okay? It's where the glands are producing whatever they're secreting that they don't really fit into the layer cell shape organization scheme. So endocrine glands just in general always do that because they don't have ducts. Exocrine glands, where their ducts are found, we can say that there's a particular layered epithelium lining the duct. But the root of the gland sometimes doesn't classify that way very well. Unfortunately, they don't really have pictures of of glandular tissue. They just have this little figure here, which is just depicting types of exocrine glands. And so, for instance, if we look at this, they don't get terribly big, but coming off of an epithelium, there's a duct here, a very simple one, like in the intestines. It doesn't look different from the epithelium it's attached to. And in fact, we usually consider those simple columnar epithelia along with the stomach or intestine lining. Oil glands are a little bit more complex than this picture makes out, but where they're actually secreting things, where this three-leaf clover looking structure is, there's a little cluster of cells there, which are not layered or anything like that. And then they go into a few different types of shapes. I don't care too much about the shapes that they're laying out here. When we get to talking about glands in the skin and that sort of thing, I'll have a few things to say about them, but more importantly, just glandular tissue, unless it's the duct of an exocrine gland, doesn't really fit well into the layer and cell shape classification system. So there's just a ninth type of tissue there. Now, while there are in fact 9 types of epithelial tissue, we are not going to look at all 9 types. We're not going to look at stratified columnar epithelium. A little bit ago, I just said that we don't really have a good slide of that epithelium. It's a very, very rare epithelium. It's found mostly where the book says right here, which I'm not going to even bother saying out loud, which is my way of saying you don't have to worry about it. But we have slides of this structure in our slide set, but the entire organ is not made up of that epithelium. It's just a little region of it. And so probably about a quarter or maybe a third of the slides that we have of that structure are actually showing us where stratified columnar is. But most of them are showing us a part of that structure with a different type of epithelium. So we don't have a reliable source for looking at that epithelium. So I'm just leaving it out completely. It's not common enough to really worry about for the sake of this class, which is actually good, because if you're going to get confused by pseudostratified, if you're going to think it's something other than pseudostratified, all you can really confuse it with is stratified columnar. So if I leave this out completely, really because it's so rare, we don't have good examples of it, that means that the pseudostratified thing, though it says it's trying to fake you into thinking it's stratified, you'll safely know is pseudostratified because it doesn't really look like any of the other tissues that you're required to look at in this class. The other ones just look completely different. So that's the way that we sort of think about organizing epithelial tissue. And it's just based on characteristics, and we come up with these different types of tissues. Connective tissues are also organized, but the organization is a little bit different. And I usually write this up on the board, and so writing it on my little note page here is going to be interesting, because I've never done it quite this way. But connective tissues are best described as being sort of within a family tree kind of structure. The term connective tissue came about because somebody was describing tissues that in fact do connect things together, like tendons that connect muscles to bones, or the tissue that connects the skin to the underlying tissue there, or tissue that holds tissue that holds muscle fibers together within your muscles, or the ligaments that hold one bone to another at a joint. Those sorts of things are connective tissues. And the name came from the fact that they physically connect things together. But then somebody discovered that there's a whole bunch of other tissues that are related to connective tissues that don't actually connect things together. Okay. Now, a lot of sources will try to tell you that they are actually connecting things together, but in fact, they're not. Unless you really want to get out in left field and talk about some very metaphorical connections. So there are things that we call connective tissues that don't really connect because they belong to this bigger family tree. So if we have a family tree, we have to have the source or the ancestor of them. And that is the embryonic tissue called mesenchyme. I'm gonna make this a little smaller so I can actually fit stuff on here. So earlier I pointed out in the book, it talks about endo, meso, and ectoderm. That's the very simplistic embryonic tissues that give rise to all the various tissues in our body. At some point within the mesoderm, a specialized kind of tissue develops called mesenchyme. And this is the source of all connective tissues. Okay. So it's the mesoderm makes mostly muscle tissue and connective tissues. A few exceptions in there, but of that, the mesenchyme is the specific subgroup of tissues that are all connective tissues. And they're in fact not all necessarily connective tissues. There are basically 4 groups of these tissues. Okay. Now I'm going to write this out, and then I've got to add some spaces in here. So if you're writing this down, hold on for a second. There's the connective tissues proper. I'm using CT for connective tissue. Doesn't mean Connecticut, even though that's what we're very close to. And then the second type is cartilage. Whoops. The third type is bone. And the fourth type is, as a group, liquid. Okay. Not that it's really a group. So laying out those 4 major types, connective tissues proper fall into 2 groups. There are the loose connective tissues and the dense connective tissues. And within that, we have a little bit more differentiation. There are 3 types of loose connective tissues, and there are 3 types of dense connective tissues. And then under cartilage, there are 3 types of cartilage. Under bone, there are 2 types of bone. And then liquid, really there's only 1 type of liquid connective tissue. So I'm going to run through these different types, and apparently they're not going to fit on the screen together. But I really wish I could do tables on this kind of page. But
you can try to tape, press tape button to separate them now. Press the tape button. Tap, not tap up.
No, that won't work.
That one, no.
No, this is just a text editor.
Oh.
So let me just run through these different types here. The loose connective tissues as a group are usually thought of as being kind of gel-like in consistency. And we use gel for a very certain specific reason when we're describing this. It's like Jell-O. Okay. Who's made Jell-O before? How do you make Jell-O? Read the box and you do it. What's the first step?
Water.
Boil water.
Yeah.
What's the second step?
Let it cool down.
No. Pour the powder into the boiling water. Yes. Then what's the next step? Then you pour it into a mold or whatever and you put it in the fridge and let it cool down. Okay. What is in the powder that you're pouring into the boiling water?
Like much.
You don't pay attention to what you have in the food that you eat?
Is it gelatin?
It's gelatin. That's why it's called Jell-O. Okay. What is gelatin? I mean, before we go there, it's more than just gelatin. There's food coloring, there's sugar, there's flavors, et cetera, et cetera. But what we want to concentrate on here is gelatin. What is gelatin? No. No, it's not phospholipid. Now, when I was doing this with the Wednesday group, I have a rather vocal student there who exclaimed emphatically that it's horse hooves. And while that's not entirely incorrect, it's a little bit oversimplified and a little bit overly dramatic. Gelatin is obtained by taking connective tissues, which you would find in horse hooves or bones or all sorts of other types of connective tissue. You boil the hell out of that stuff, okay? And you break the tissue down completely. And then at the top of this stuff, and I've never actually done this, so I don't know if it's actually at the top, but let's pretend. At the top, a particular type of molecule rises up, and you can skim that off the top of the stuff and clean it, purify it, whatever, and that's going to end up being gelatin. Okay. Now, what it is it's called collagen. You've heard of collagen, right? Okay. With the Oscars last night, you might have seen some collagen injected in the lips and that kind of thing. And oh my God, did you just email me? But anyways, when you do that, when you boil the hell out of tissue and you basically remove that layer that's collagen, that process is called rendering. Okay, and it's a way to separate out different components of tissues that coming out of the slaughterhouse or something like that. When you do that, when you boil the hell out of that tissue, you're going to denature the proteins, and collagen is a protein. And then as it's removed and separated and prepared, the proteins are going to settle into a new kind of shape after they've been denatured. And that new shape is what we call gelatin. Okay. It's really hard, I imagine, to sell a package of rendered collagen out. So they call rendered collagen gelatin. That's where it gets its name. Now, probably they call it gelatin more because not somebody said, hey, we're not going to be able to sell rendered collagen out. Let's call that something else. Gelatin was probably the name that was around before we even started talking about collagen. Okay. They just went through this process. Somebody discovered that you can do this to get stuff out of connective tissues, and they might have used that to make jellies and jams or something like that. And obviously jelly and gelatin are related terms. So there's some history to where it actually comes from, and we're not going to get into that. But the reason I'm talking about this is that the loose connective tissues have that kind of Jell-O consistency because they're actually basically Jell-O. Okay, the collagen hasn't been rendered. It's collagen in its natural form, but it's a concentrated collagen solution. Okay. Now, why do you boil the water before you add the gelatin to it? Have you ever tried to make Jell-O starting with cold water?
No.
It doesn't work.
Why not?
Does it dissolve?
Yeah, you need to heat it up to dissolve it. You saturate the solution with the gelatin. Okay? And then when it cools, that supersaturated solution turns into this gel-like consistency. And that's essentially what it is. Just a high concentration of collagen in a solution is going to have that kind of consistency to it. Okay? And that's essentially what we have here. So there are three types of tissue. The primary type is areolar connective tissue, and CT for connective tissue. Now, this is sort of the go-to example of a good connective tissue. We find it everywhere throughout the body because it's always adjacent to epithelium. Now, I went through talking about epithelia a little bit quickly, and there's something I didn't mention about epithelia, which is kind of important, especially when we're talking about this. Now, I like to use this brick wall as an example of an epithelium. Okay? So we have bricks in this wall that are all packed closely together, kind of like the cells in an epithelium. And I skipped over, but in the chapter in the book, they talk about the adhesion points that hold epithelial cells together. So sort of like the mortar in between these bricks, there are things that actually hold the cells together. And there's not really any space for stuff to fit in between those cells. So just like these closely packed bricks in this wall, cells in an epithelium are very closely packed together. Blood vessels cannot get into an epithelium. Epithelial tissues are, by definition, avascular, meaning they have no blood vessels. And so blood has to be supplied from the adjacent tissue. So let's imagine this electrical conduit right here that goes from this switch box. Pretend that's a blood vessel. Okay? So blood's coming in adjacent to the basement membrane of the tissue. And the nutrients from the blood will diffuse through the tissue. Wastes from the tissue will diffuse back to the blood. The thickness of stratified epithelia is always limited by their access to blood. Or I shouldn't say always limited, but basically limited by their access to blood. You can't have blood diffuse more than a certain number of layers through cells. And there's not some golden number for that, but there's a limit to how much things can diffuse from the source of blood into the tissue. But areolar connective tissue is where we find those blood vessels. Okay? It has cells scattered around, but
it's not actually like the blood tissue. It's like the surrounding.
The blood vessels. Oh, you mean diffusing?
Yeah, no, the oral. That's not the actual. Surrounding.
No. It's not the blood vessel wall.
Yeah.
It's the tissue is.
It's the part that makes the. Is it essentially what makes it go where it is intended to go?
I wouldn't exactly describe it that way. I'd say more, and this is kind of a good definition of a lot of connective tissues. There are some cells here and there, but then there's a lot of extracellular space. And that extracellular space is filled with collagen fibers and a few other types of fibers. But because there's all this extracellular space, we're also going to find the blood vessels. Okay? And so by having a loose connective tissue right next to the basement membrane of an epithelial tissue, so if this is areolar connective tissue space right here, we can get blood vessels in right next to it. So while we can't put the electrical wire through this concrete block, we can have it laying right next to it because there's some space beyond it. So the blood vessel is in there. And actually, let me just switch to the book quickly. They should have a somewhat representative picture of this. Yeah. So this is just a drawing showing a few different types of cells and fibers filling in some of the extracellular space, but then all of this kind of, I don't know, pale yellow, green color, whatever that is, the background, that's empty space. And then over here, we're looking at an actual slide of some tissue. And they're pointing at some adipose cells and other kind of cells that are kind of in this tissue. But probably, yeah, maybe. This thing right here, this is probably the wall of a blood vessel, okay? So it's in the space here, sort of passing through. I wish they'd kind of had a version of that in the slide here. And I'm just grabbing some images from a Google search. Not a good example. Oh. Shoot. Did not go where I wanted to. Didn't quite get as big as I was hoping, but this right here is definitely a blood vessel. This is a blood vessel. This is a blood vessel. And they are very close to the basement membrane of this epithelium right here. That's what we usually associate with areolar connective tissues, that there's space within the tissue for blood vessels to get in. So they're found everywhere. The next type, which is fairly similar, is called reticular connective tissue, which is mostly associated with what we call the lymphatic system. The go-to example of that is the lymph node, which is what the slide we have in our slide box is. But then another example of reticular tissue that I want to point out is red bone marrow. And I point that out because red bone marrow is what makes the cells that make up blood, and that's how blood is going to be related to the connective tissues. It's not directly made from mesenchyme, but it's made from red bone marrow. The third type is adipose tissue, which is still considered gel-like, but it's not exactly like the other two. It doesn't have necessarily as much extra space in the extracellular tissue, extracellular matrix. It can, and there can be blood vessels passing through that tissue also, but really it's taken up mostly with these adipose sites, the fat cells. Under the microscope, adipose sites don't stain because lipids don't stain. And so it looks like it's got empty space in it, but it doesn't really. But it's still kind of gel-like in its consistency. Not for the same reason, not for the jello reason I talked about earlier, but rather that adipose, or fat, at body temperature has that kind of consistency to it. The dense connective tissues, they're all basically defined by having a lot of collagen to them. The usual example of a connective tissue is a tendon. And a tendon is, in fact, a type of dense connective tissue because it's really just all collagen fibers that go from a muscle to a bone. And the stress that the muscle puts on the bone as it's moving is specifically along the lines of those collagen fibers. Now, there's two major types of dense connective tissues. There's dense regular, and dense irregular. The difference between these two is really about how the collagen fibers are oriented. Tendons are dense regular connective tissue because all of the collagen fibers are oriented parallel to each other. They go from point A to point B, from the muscle to the bone. So if I grab a model of muscles, the white stuff that's painted on here are the tendons. And I was hoping to grab a leg one because there's some good examples right there. But you can see all of these tendons, say, going across the superior surface of the fingers, or maybe even better, here across the wrist and heading towards the inferior surface of the fingers. Those are all collagen fibers that are oriented from the muscle, which is right here, to the bone that it's connected to, which is down here. And so when this muscle contracts, it pulls on the tendon in that one very linear direction, so all the collagen fibers are oriented that way. That's what regular means. Irregular just means that the collagen fibers are oriented in a very different way, or they're not parallel to each other. They're kind of going all over the place. And a good example of where dense irregular connective tissue is, or dense irregular connective tissue is in the skin. The epithelium part of the skin, the epidermis, is connected to the tissues beyond it by what we call the dermis of the skin. And that is mostly dense irregular connective tissue. Now you can pull on your skin and it has the strength, and you don't just yank it off your body because of all the collagen fibers in the dermis. But you can be pulling on the skin and there's going to be stress along those collagen fibers in an infinite number of different directions possibly, okay, depending on where exactly you pull. So there has to be collagen fibers oriented in every possible direction to deal with the stress that you're putting on the tissue. So they're going to be irregularly organized. They're going to be all mashed together. The third type is kind of like regular, but it has a lot of another type of fiber in it besides the collagen. It has a lot of elastic fibers in it. So that means that it can stretch out and then sort of snap back to its original shape. Now, a good example of where we find this tissue is in the lungs. When you breathe, muscles expand your rib cage, which sort of stretches your lung out, and air rushes in and it fills it up, kind of like an air balloon. But then, just like a balloon, if you let if you keep the end open, the air will rush out because the rubber of the balloon is going to recoil and push the air out. That's what happens in your lungs. You stretch the lungs out and fill them with air, and it's the recoil of the elastic tissue really squeezing the lungs back to their smaller size, pushing the air out. That's an elastic, dense elastic connective tissue. Now, the example that we're actually going to see under this microscope is in the wall of the aorta, the blood vessel that blood is pumped into from the heart. So each time the heart beats, there's a really forceful ejection of blood into the aorta. So it's got elastic tissue in it, so it can suddenly increase in size to accept that ejection of blood. And then it squeezes back down and pushes the blood on into circulation from that point. So that's what we're going to see in the microscope. Now, cartilage falls into three different types. There's hyaline cartilage, there's fibrocartilage, and elastic cartilage. Now, elastic cartilage is not the same thing as dense elastic connective tissue. They both are elastic, but they're different. One thing that defines cartilage is that it's specialized in the cells and some of the molecules found in its extracellular space. One of those molecules that's very important is called chondroitin sulfate. I like to describe chondroitin sulfate as kind of a big hairy molecule. Okay? Who has pets? So you're probably familiar with little dust bunnies collecting in the corners that is made of pet fur. Okay? And the way that those hairs collect together, you get this little cluster of, you know what I'm saying. The dust bunnies are collecting there. That's kind of what chondroitin sulfate does. It's these big molecules, and they all kind of mesh together to give cartilage a sort of rubbery consistency. Now, elastic cartilage has chondroitin sulfate, and it has a lot of elastic fibers in it too. Fiber cartilage has chondroitin sulfate, and it has a lot of collagen fibers in it, or fibers in it too. And then hyaline is sort of your basic cartilage. It also has collagen in it, but not nearly as much as fiber cartilage. Now, in describing these, I want to tell you where these are found, and therefore give you some examples of what they're like. I want to start at the bottom of the list because it's easiest to start there and then work our way up. Elastic cartilage is really only found in two places that we care about in this class. The first one is very easy. It's your external ear. Except for the earlobe, if you don't have an attached earlobe, all of the rest of your ear, that really rubbery kind of feeling thing that you can bend, fold, and mutilate all you want, and it'll snap back to its original shape. That's elastic cartilage. And that's a really good example of what it means for tissue to be elastic, that you can do all sorts of things to it, and it'll always go back to its original shape. That's the external ear. The other place where it's found is in what's called the epiglottis. This is a little flap of tissue that covers your lower respiratory tract when you swallow. So when you take a swallow, a little flap of tissue folds down so that whatever you're swallowing goes down the esophagus instead of the trachea, your windpipe. It sits right about here, kind of at the top of your throat. It's usually in an open position so that air can move in and out of your lungs all you want. But when you swallow, it folds down. And then the elastic tissue in it allows it to pop right back up and keeps it in the open position. That happens a lot throughout the day. Okay? You're producing saliva in your oral cavity and you're producing mucus in your nasal cavity throughout the day. And all of that fluid has to be sent down your esophagus eventually. So you're always kind of swallowing that stuff. And so the epiglottis is folding back and forth all the time. And it's elastic cartilage to give it that ability. Just like your ears, you can fold and bend all you want, and it's always going to snap back. Okay, and when you're 80 years old, your ear's still going to be elastic like that. The epiglottis is always going to be elastic like that. If it ever lost its elastic ability, then you would stop breathing. So it's got to be able to do that throughout your entire life. Fiber cartilage is primarily found in structures that we see in this model right here. The actual rubber in this model, and like I said, cartilage kind of has a rubbery consistency to it, is the fiber cartilage. So there are these pads in between each vertebral bone in the spinal column. And then there's a pad right here where the two pelvic bones come together at the pubic arch, which is called the pubic symphysis. Those are made out of fiber cartilage. There's also fiber cartilage in the knee, which I have another model here. So if I move the tendon and the patella kneecap out of the way, in this model we can see a little piece of rubber right here where the femur sits on top of the tibia. Those are called the medial and lateral meniscus. And they're made out of fiber cartilage also. All three of these places that I mentioned, the intervertebral discs, the pubic symphysis, and the meniscal tissue in the knee, all are where we bear most of our body weight. And fiber cartilage can absorb a lot of that compressive force and not be destroyed. The fibro part of fibrocartilage is basically that there's a very dense network of collagen fibers around the outside of the cartilage pad. And so when you press down on it, it won't sort of squish out, sort of like if you took an ice cream sandwich and you push down, the ice cream's going to come out on either side. But if you had something holding the ice cream in the ice cream sandwich, that's not going to happen. I kind of came up with that off the top of my head. You get the point there? Now, there are times when that doesn't work properly. Herniated disc is one of those times. So here, this little red thing sticking off to the side of the intervertebral disc, that's a little bit of the cartilage that's pushed out, herniated out, past the fibrous enclosure. That's a bad thing. And something like that's usually pushing on the adjacent nerve, which creates a lot of pain. But So sometimes it doesn't quite do its job, but the point is that it can absorb a lot of that compressive force from our body weight on the length of our spine, where the pelvis meets in the front, and at our knees. That's fiber cartilage. So I've just mentioned 5 different things. Elastic cartilage is in the external ear and the epiglottis. Fiber cartilage is in the intervertebral discs, the pubic symphysis, and the menisci, I guess that's the plural, in the knees. Other than those 5 places, anywhere else that we have cartilage, it's hyaline cartilage. That's why I started at the bottom. Those 5 things are easy to describe, and then everything else in your body that's cartilage is hyaline cartilage. There's cartilage at every movable joint in your body. There's cartilage keeping the lower respiratory tract open. The larynx and the trachea and the bronchi in your lungs, they're all lined with cartilage. The ribs are connected to the sternum by cartilage. All of those things are hyaline cartilage. Anywhere else that you come across cartilage in the body, it's probably hyaline. Other than the 5 things that I mentioned there. Yeah.
Are the float up cartilages, are they like hard bone? Are they like fluid cartilage? Are they just cartilage, right? The float ups, the along the bone.
No, the ribs themselves are bone. It's where everything but the floating ribs are connected to the sternum. They're connected by cartilage.
Okay.
And we have these wonderful little models of that. Whatever. If you look at one of these articulated skeletons, all of this stuff here that's going from a rib to the sternum, that's cartilage. And then there are 2 ribs that are sort of in the back, kind of covering the kidneys, that are not connected by cartilage. But everything else is cartilage. And I don't know where those models went to. But okay, which brings us conveniently to bone. There are 2 types of bone. There's compact bone and spongy bone. Now, just like cartilage is known for having a very rubbery consistency from chondroitin sulfate and similar molecules, bone is known for having a very hard consistency because of calcium phosphate crystals. The main one being hydroxyapatite. Bone cells secrete what we call bone matrix that lays down the extracellular component of bone, which has these calcium phosphate crystals, hydroxyapatite. What I have here is an actual bone. Okay. One of my colleagues took it home, put it on his bandsaw, cut it in half, sliced off one end of it, and mounted it on this lovely plaque. Okay. What you're looking at here is pure bone matrix. This bone obviously is not in somebody's body anymore, and so it's not living bone. And since it's no longer living, it's all of the cells and blood vessels and nerves that were part of the bone before have degenerated away. And all that's left over is the bone matrix, the extracellular part of the bone, which is this hydroxyapatite stuff. It is very durable. Okay. The compact bone is the solid wall that we can see along the shaft of this bone. And really, it continues and covers all of the bone. So even down at the edge, the outer covering is still that compact bone. Gives it kind of a smooth outer covering. Spongy bone is what we see in the center, okay? Mostly down here at the end. So it looks like a sponge, like a loofah sponge or something like that, which is why it's called spongy. It's still hard bone. Don't pound on the spongy bone like you can pound on the compact bone part here. If you do this to the spongy bone, it'll start to break. Please don't do that. But it's still that same sort of calcium phosphate crystal structure, which gives a lot of strength. Spongy bone is a lot lighter than compact bone, and so aside from the big weight-bearing bones in our body, like in our legs and arms and that sort of thing, our bones are mostly made up of spongy bone tissue. It's hard, it's just very light because it looks like a sponge. It has a lot of extra space in it. That extra space is filled with bone marrow. Okay? Not necessarily red bone marrow. Okay? Bone marrow comes in two types. There's red bone marrow, which is a type of reticular tissue, and there's also yellow bone marrow, which is basically adipose tissue. A newborn basically has red bone marrow everywhere in all of their bones. But then as we get older, a lot of that red bone marrow converts to adipose tissue, to yellow bone marrow. And it's really kind of a energy storage compartment. So if you need energy, you have fat tissue there to break down if you need it. But it's mostly, bones are mostly made up of spongy bone. And when we get to talking about bones, which will actually be next week, we'll look at the structure of bones in a little more detail. And then finally, we have liquid tissue. Now, some sources might talk about multiple types of liquid tissue, but really there's just blood. If you find a source that's talking about liquid connective tissues and they mention lymph, I don't really buy it. Lymph, well, a tissue by definition is going to have cells in it. And any cells that you find in lymph are white blood cells. And they're called blood cells for a reason. They're blood cells, so they're from the blood. They leave the blood and move into the lymph. So lymph isn't really a tissue by itself. It's really an amalgam of things from elsewhere. So really there's only one liquid tissue, which is blood. So that's kind of the way to organize connective tissues. Now, about 15 minutes ago, I hesitated for a little bit and looked at the clock because it was sort of the halfway point, time to take a break. I decided not to take that break, and I decided not to tell you about that because I wanted to power through and finish this up. The reason I decided not to take a break is that I'm now done talking about the stuff I want to talk about in this material. What we're going to do for the rest of today and then continue on Wednesday, because it's a little too soon to shift all the way to the skeleton next time, is to look at this stuff under the microscope. So in doing that, what I'm going to have each of you do is look at a particular tissue under the microscope. It'd be great if you could look at all the tissues and get to know what they all look like under the microscope, but it's not really a realistic use of our time. So each of you is going to be in charge of looking at a particular tissue under the microscope. I'm going to then, or no, in doing that, you're going to capture images of that tissue. And I'm going to post that online where you can get to it. So right now there's a link in Blackboard that says microscope image capture. So if you click on that, you'll go to this page here, which is just a cloud-based folder where I've stored images that were captured last semester by my MP1 students. For each of these, I have the name of the tissue, the objective lens that we used to capture it, and the organ that this particular tissue comes from. Okay, so this is transitional epithelium at 40x objective using the ureter. Here's transitional epithelium at 10x objective using the ureter. Here's transitional epithelium at 4x objective from the ureter. Just all three magnifications are captured there. You're going to be assigned one of those tissues, and your job is to find it under the microscope and get pictures of it captured with the digital scope up here. That's what we're going to be doing with the rest of today, and if you don't get it done today, on Wednesday we'll continue with that. Hopefully on Wednesday we'll be completely done with all the tissues by about the midway point, and we can start talking about the skeleton so that we don't have to, the skin and the skeleton, so we don't have to cram that all into next Monday's class, but we'll see how it works out. So to do that, this scope up here works a lot like the scopes there. I'm going to have each of you, I'm going to assign you your tissue, and each of you is going to find it on your scope. I want you to show me that you found it on your scope, which will help me understand if you need help using scopes, and hopefully we'll get to the point where everybody can use a scope. And then when I've seen your tissue and I'm satisfied that you're using the scope fine, you're going to come up here and capture that tissue with this microscope. And then we'll save it and it'll be on Blackboard there for everybody to look at. So let me just stop.